Okay. So again, welcome back to the Excelsior Academy. So now, in the previous video, we were talking about the sodium channels. Now we are going to discuss of another channel that is called potassium channels. Okay. So let's talk about potassium channel. So potassium channels, so they are widely distributed. Now, who can tell me why they are the widely distributed ones? Why they are so widely distributed? Because all your nerve activities, all your motor functions, everything is dependent on your potassium channels. Okay, so whatever activity you do, even while like speaking, seeing, sitting, whatever we do activities, in each and every function your potassium channels are involved. Okay, so they form the potassium selective pores that span the cell membrane. So here, see, remember in the case of potassium channel also we were talking about the selective pore which actually decides that only the Na plus should go. Okay. In the same way, the potassium channel is also having a selective pores which is called selectivity filter. Now here this filter is playing a very important role because there are many other molecules which are of the same size, having the same charge, can actually go and bind, and they are called the potassium blockers. Okay, so these selectivity filter, what they do, they make sure that they are making good interactions with the potassium ions. So let's see how it is doing. So when the when the potassium voltage gated channel opens, membrane potential from minus 70 millivolt, it becomes to more negative. So what is happening? It is becoming more negative, whereas in the case of sodium, it was becoming less negative. It was going towards the positive, and in this case, it is going more towards the negative. Now, why this is happening? If you remember the figure, in that case, Na plus was coming from outside to inside. Okay. Now, in this case, you can see that the K plus is going from inside to outside. So it means what? Loss of positive charges, and hence it is becoming from minus 70 to minus millivolt. Is this much clear? Okay. So here also the role is played by the neurotransmitter. Can you give any few examples of neurotransmitter? Any one example of neurotransmitter? There are certain amino acid neurotransmitters also. Any good example?
Acetylcholine, ma'am.
Very good. Yes. So we have got the acetylcholine.
Dopamine, ma'am.
Yes. Dopamine is there.
Dopamine.
Yes. And then we have got serotonin also. Okay. Very good. Now,
Yes.
one small assignment from here. How do you synthesize neurotransmitters? Okay, I mean in the body, how it is being synthesized. So you know that acetylcholine is from acetyl plus choline. Which enzyme is involved and which enzyme is it is degrading. Okay, same way, from where dopamine is made, which enzyme is synthesizing and which enzyme is degrading or breaking. Same way with the serotonin. So just remember these few, and it will be useful because in membrane potential we are going to study about these. So it's going to be useful. So what is this assignment? You have to just write the enzyme which is involved in the synthesis and degradation of acetylcholine, dopamine, serotonin. And many other neurotransmitters are also there. You have got GABA also. Okay, gamma aminobutyric acid. So just take 4-5 neurotransmitters and study their synthesis. Anyways, I'll take that too. But before that, please study. Okay. Now the structure. So in the sodium channel, we saw that it has got the 4 subunits, right? It had the 4 subunits with each subunit having 6 segments, correct? Now in this case, it is a tetrameric structure. Okay, so you can see 1, 2, 3, and 4 helices are there. I think someone, okay. Yeah. So it has got the tetrameric structure. So it means that it has got the 4 identical protein subunits, and then they associate to form a C4 symmetrical structures. So what does it mean? Symmetrical means from each 4 sides, it is going to look same. Okay. Now, when they come together, all these 4 subunits come together, there is a central ion, there is a central ion conducting pore. Now, what is SF? SF stands for selectivity filter. Okay. So now what we have said it till now. So slowly we'll go, no hurry. So we have got 4 subunits, again, identical protein subunits. It's a tetrameric structure. It forms a symmetrical structure. In the center, you have got the ion conducting pore, and we have got the selectivity filter. Okay. Can you see over here the pore has been shown? So you have got, this is a pore. So this is now actually a closed structures like this. So when your K plus comes, it goes through the selectivity filter. If it is correct, then it goes and passes, and then the channel gets open up. So this is your inner helices, outer helices, pore helices, all these are there. So can you see this structure and this structure is actually your pore helices, which is helping in the selection of the K plus ions. Okay. Okay. Now what you have to remember over here is that the potassium ion channels, they actually remove the hydration shell from the ion when it enters the selectivity filter. So remember in the previous lecture when I said that K plus and Na plus, they have got the water hydration shell. So in the case of potassium channel, it is removed, whereas in the case of Na plus, it is not removed. And that's why the lumen of the pore of the channel is small in case of K plus and it is large in case of Na plus. Okay. So once again, it removes the hydration shell from the ion whenever it is entering the selectivity filter. Now, what is important? This is very important point. Your selectivity filter is formed by 5 residue sequence called TVGYG. What does it mean? It is the threonine, valine, glycine, tyrosine, and glycine. Okay. Term this signature sequence which within each of the 4 subunits. So your selectivity filter is having a 5 residue sequence, that is threonine, valine, glycine, tyrosine, glycine. Please by heart. Okay. Now this is the signature sequence within the loop between the pore helix. So when I showed you the pore, so all these 5 residues are present forming the selectivity filter to identify the correct ion is incoming or not. Okay. Now, this signature sequence is highly conserved with the exception that valine residue in prokaryotes potassium channel is often substituted with isoleucine in case of eukaryotes. So this much you have to remember. So in case of prokaryotes, it is valine. In case of eukaryotes, it is isoleucine. Okay. Is it clear this much? Is it clear?
Yes, ma'am.
Okay. So slowly we'll go because so many things are there to remember. Now, look at the structure over here. So, the 4 sets of electronegative carbonyl oxygen atoms are aligned toward the center of the filter pore. So, if you see the structure, so this is the blue color helix which I was talking about, that it forms a pore helix. Now, can you see the loop over here? It has been shown in the bracket. So, these. So, these are the amino acids whose carbonyl group, if you see the peptide bond, it is like this, right? We have got the peptide bond. So the carbonyl group of the peptide bond is aligned. So oxygen from the carbonyl group of the peptide bond is aligned like this. Okay. So toward the center of the filter pore. Now, what you have to understand here is the distance between the carbonyl oxygen and the K plus. So oxygen and the K plus distance. This distance is same between the water oxygens in the first hydration shell and a potassium ion. So we know that when K plus hydrogen ion, okay, so when there was a hydration shell around the K plus, so this distance is now replaced by the this oxygen of the carbonyl group. So what they are doing actually, they are removing the entire hydration. Okay, so water is removed now, and this interaction of O minus and K plus has been replaced by the O minus K plus, where oxygen is of the carbonyl group. Getting the point? So the water is removed, now carbonyl is there, still maintaining the same distance. So this becomes the energetically favorable route for desolvation of the ions. It means removal of the water. Is it clear till here? Is it clear? Take your time, no problem. Okay. Okay. One second. Okay. Okay. So that was all about the structure. So if you have any doubts, you can ask me. So now what is happening is, your K plus ion comes, water has been removed, okay, and this is removed by this carbonyl oxygen atoms, then it moves inside and then it interacts, and then what happens? Can you see the figure over here? Suppose these are your K plus carbonyl groups are there, and then when this making interaction is correct, the channel from here it gets open up, and then it transfers the K plus ion. Okay, so that was all about the potassium channel. Since we studied a lot in the case of sodium channel, so studying potassium becomes very easy. Okay, so thank you for listening.